Hello, my dear NEET aspirants. A very warm welcome again to your amazing session of Power Hour with your zoology mentor and guide, Dr. Ami Bayani. Today, we are going to have a discussion on some of the most important topics of human physiology. We are starting with Unit 5 of 11th Standard NCERT. And in human physiology, today we are going to talk about some conceptual part of breathing and exchange of gases, body fluids and circulation, and locomotion and movement. Okay. So we are going to talk about lungs, how they function. We are going to talk about heart, how it beats, and we are going to talk about muscles, how they work. So let's get started. Okay. In breathing, as I told you, we are going to talk about transport of gases, oxygen dissociation curve. In body fluids and circulation, we are going to talk about the conducting system of heart, cardiac cycle, and a little glimpse of ECG. In locomotion and movement, we are going to talk about what is a sarcomere, its structure, what is Z-line, what is A-band, I-band, the physiology of how muscle contraction takes place. Okay. Now, starting with our topic of breathing and exchange of gases, we have studied that when we are talking about respiration and breathing are two different terms. What is respiration? Respiration is basically a process where oxygen that we have taken in goes to the tissues, and in the tissues, the carbohydrates are broken down, or respiratory substrates are broken down to release energy, which we use for all the various activities of our body. So what are the steps of respiration? The first step of respiration is breathing, which is also called as pulmonary ventilation, as given in the NCERT. However, this is the term that most of us tend to neglect. So make a note of it that breathing is also called as pulmonary ventilation, which includes two steps, inspiration and expiration. Inspiration, where we take in the oxygen rich air, and expiration or exhalation is when we release carbon dioxide rich air. Followed by diffusion across the alveolar membrane. Now this diffusion across the alveolar membrane is also given a term called external respiration. So transport of gases, followed by diffusion of gases between blood and tissues, followed by cellular respiration. Now we know how air enters into our lungs and how it is exhaled out because of the pressure gradient that is created. Herein, we are going to discuss about the external respiration, that is diffusion across alveolar membrane, transport of gases, diffusion between blood and tissues, and how it takes place. So let's begin. Now, when we are talking about transport of gases and diffusion of gases, that is external respiration, transport and internal respiration, if you are talking about external respiration, where does exactly, or which is the primary site where exchange of gases takes place? The primary site where our exchange of gases takes place is mainly the alveoli of the lungs. Okay. In the alveoli of the lungs, or surrounding the alveoli of the lungs, the blood that reaches is deoxygenated blood. If you just recap your circulation chapter where you studied about pulmonary and systemic circulation, if you remember, from the right ventricle, deoxygenated blood goes to the alveoli via the pulmonary artery, from where oxygenation takes place, and oxygenated blood comes to the left atrium. From the left atrium to the left ventricle, from the left ventricle, then the systemic circulation starts, taking the blood to the tissues, from there back to the right atrium. Now, when the oxygen has to enter into the blood, there are various factors which will promote the movement of oxygen from the lungs or from the alveoli into the blood, and carbon dioxide from the blood into the alveoli. What are those factors that favor this movement? So the factors which are favoring the movement of oxygen from the alveoli into the deoxygenated blood and carbon dioxide from there into the alveoli are three: partial pressure of gases, solubility of gases, and thickness of membrane. How does this work? We will look into it. But remember, one more important parameter and concept which can come as MCQ is that exchange occurs how? It occurs via the process of diffusion. Primary site alveoli, and process occurs via diffusion. Let us take into consideration all the three parameters one after the other. The first one, partial pressure of gases. Now, what do we mean by partial pressure of gases? When we are talking about our atmospheric air, it is nothing but mixture of gases. That air has a particular pressure. If air is made up of multiple gases, every gas will exert its own pressure, contributing to the pressure of air. So that pressure of a particular gas in the mixture of gases is nothing but the partial pressure of gases, which we denote in the parameter of millimeters of mercury. Now here we are going to talk about only two gases, which are respiratory gas, which are the main respiratory gas. Can you tell me which are the two main respiratory gases? Yes, oxygen and carbon dioxide, correct. Now when we are talking about the oxygen, one after the other we'll talk about. When you're talking about oxygen, the oxygen partial pressure in the atmospheric air is 159 mm of Hg. While from the atmosphere it reaches to alveoli, in the alveoli it is 104 mm of Hg. If we talk about the deoxygenated blood, in the deoxygenated blood it is 40 mm of Hg. If we are talking about the oxygenated blood, in the oxygenated blood it is 95 mm of Hg. In the tissues which have undergone metabolism is 40 mm of Hg. Do not worry, we are going to talk about this again when we talk about transport of gases, just giving you an overview. Again, if we are talking about carbon dioxide in the atmosphere, carbon dioxide is 0.3 mm of Hg. In the alveoli, what do you think? Will the deoxygenated blood have more of carbon dioxide, or the alveolar air will have more of carbon dioxide partial pressure? Yes, it will be the deoxygenated blood. Very good. So in the deoxygenated blood, the partial pressure of CO2 is 45 mm of Hg, whereas in the alveoli it is 40 mm of Hg. In the tissues which are undergoing metabolism, it is 45 mm of Hg, and oxygenated blood is 40 mm of Hg. Now if you just pay attention to the alveoli and the deoxygenated blood, in the alveoli if you see, partial pressure of oxygen is 104, in the deoxygenated blood is 40 mm of Hg. So you see, we just spoke that the exchange takes place via diffusion. Diffusion is movement from higher pressure to, yes, lower pressure. So oxygen will go from the alveoli into the blood, and carbon dioxide, if you see, in the blood is 45, in the alveoli is 40, so it will move from the blood into the alveoli. So the partial pressures are favoring the movement of oxygen from the alveolar air into the deoxygenated blood, making it oxygenated, and carbon dioxide from the deoxygenated blood into the alveoli. Is this clear? Moving ahead. The second factor which affects the movement or the exchange of gases is solubility of gases. Now the more the soluble the gas, the faster it is to diffuse. Okay. Now there's a statement in NCERT which says that the solubility of carbon dioxide is 20 to 25 times higher than that of oxygen. Hence, the amount of CO2 that will diffuse across the membrane per unit difference in the partial pressure is much higher than that of oxygen. Do not worry, just give me a second. So let us see what it means, okay? So solubility of CO2, 20 to 25 times higher. Now this can come as our MCQ, okay? Than that of oxygen. So, as we just saw here, the partial pressure of oxygen is 104 mm of Hg in the alveoli. In the deoxygenated blood is 40. How much is the difference? 64 on an average mm of Hg. But if you see for CO2 partial pressure in the alveolar air is 40 mm of Hg, and that in the deoxygenated blood is 45. How much is the difference? 45 and 40. So, how much is it? Just 5 mm of Hg. Now think, here there is just 5 mm of Hg. Here there is 64. Why such a difference? Are you able to guess? Yes, we just said the solubility of CO2. Because CO2 is 20 to 25 times more soluble, the amount of CO2 that will diffuse from where to where? Diffuse from the blood to the alveoli in 1 mm of Hg difference will be much higher than the amount of oxygen that will diffuse into the blood. Hence, the difference here, even for carbon dioxide, if it's just 5 mm of Hg is enough for the amount of CO2 to diffuse out of the blood. But for oxygen, it will require a greater difference. Are you understanding? So again, the solubility of gases are favoring movement of CO2 out in the alveolar air from the blood and movement of carbon dioxide into the blood. Is that clear? Cool. Moving ahead to the third parameter, which is thickness of membrane. Now what membrane are we talking about here? We are talking about a membrane which is this membrane called as the respiratory membrane, or we call it as the diffusion membrane. What membrane? We call it as the diffusion membrane or the respiratory membrane. Now this membrane is made up of 3 layers. What are the 3 layers? The 3 layers are, one, you see alveoli. If you remember in the structure, what is the alveoli made up of? Which epithelium is it lined by? Even structural organization. Which epithelium? Yes, it is simple squamous epithelium. So you have the simple squamous epithelial lining of the alveoli. We have the endothelium of the blood vessel. Again, my dear children, if you remember, it is again squamous epithelium. So, the epithelium of the alveoli, one membrane, layer of the membrane. Second is the endothelium of the blood vessel, the second layer of the membrane. And third is the basement substance. Now, what is this basement substance? The basement membrane supporting the alveolar epithelial cells and the basement membrane supporting the endothelium of the blood vessels, together called as the basement substance. Again, a very important MCQ. What is this made up of? This diffusion membrane made up of 3 layers: squamous epithelium of alveoli, endothelium of alveolar capillaries, and the basement substance. Okay. Now, the thickness of the membrane, what do you think? Will it matter? Yes, it will matter. How? The thicker, the faster the diffusion, or the thinner, the faster the diffusion? The thinner, the faster the diffusion. So, this membrane which is present, the diffusion membrane, is less than 1 millimeter thick. Now imagine, to call it thick is also an irony because it is less than 1 millimeter thick. Again, you have to remember, again favoring an easy exchange of gases. Is that clear? So exchange of gases, 3 parameters on which it depends upon. One is the thickness of the membrane, the partial pressure of gases, and third, the solubility of gases. Is that clear? Okay. Moving ahead to the further understanding. Now, when we move ahead to further understanding, we just saw that one of the factors which is there is one of the steps which is there after inspiration is exchange at the alveolar level by diffusion. What happens further to the gases? Let us have a look, that is transport of gases. When we are talking about transport of gases, transport will be of oxygen and of carbon dioxide. How does this take place? Now we just saw that partial pressure of oxygen in the alveoli and the deoxygenated blood is such that it will favor movement of oxygen into the blood, right? Once oxygen moves inside into the blood, how is it transported? The medium for transport is blood. How in the blood it is transported? Let us see here. Once oxygen enters into the blood, 97% of oxygen combines with hemoglobin, the red colored pigment of the RBCs, to form what is called as oxyhemoglobin. Now remember, very, very, very important, I'll make a note for you all here. Now this binding is reversible. What do we mean by reversible? Reversible meaning that it can be easily broken down and it can go in the other direction, wherein oxyhemoglobin can be easily broken into hemoglobin and oxygen can dissociate from it. So this process where the hemoglobin carries oxygen remember, is not oxidation, it is oxygenation. What is it? Oxygenation. So 97% binds with hemoglobin in a reversible manner. Very, very, very important. To form what is called as oxyhemoglobin. And 3% is in the dissolved form in the plasma. Thereby, the oxygenated blood has 97% oxyhemoglobin, 3% dissolved form of oxygen, and the partial pressure of oxygen in the oxygenated blood is 95 mm of Hg. Now, this blood with oxygen goes to the left atrium, from there to the left ventricle, from there is carried to the tissues. Now when it comes to the tissues, the partial pressure of oxygen in the arterial blood or the oxygenated blood is 95 mm of Hg, and tissue cells and tissues which are doing metabolism will have a partial pressure of 40. Why so? Can you think of it and tell me why so? In the chat, come on, quickly. Why will this have Yes, because it is doing metabolism. So since it is doing metabolism, it is utilizing oxygen. Since it is utilizing oxygen, the partial pressure of oxygen is less. And the partial pressure of oxygen in the arterial blood is high. Again, remember, how does it move from one side to another side of across the membrane, the gases? By diffusion. So via diffusion, oxygen will go from the arterial blood into the tissues, and the oxyhemoglobin will have a dissociation where oxygen leaves hemoglobin and enters into the tissues. Is this much clear? So that is transport of oxygen. Now let us look into carbon dioxide, another friend of oxygen. Now what happens is, because the tissues are doing metabolism, the amount of carbon dioxide is high. As the amount of carbon dioxide is high, its partial pressure is 45 mm of Hg, and that in the oxygenated blood is just 40 mm of Hg, favoring diffusion of carbon dioxide from the tissues into the blood, making the blood now deoxygenated. That is, less of oxygen, more of carbon dioxide. Once carbon dioxide enters into the blood, where does it go? Majority of it enters into the RBCs, its new friend. Now once it enters into the RBCs, now in the RBCs it combines with water in the presence of an enzyme, the fastest acting enzyme. Do you remember last power hour session we discussed enzymes? I told you the fastest acting enzyme in the body is, yes, carbonic anhydrase. So in the presence of carbonic anhydrase to form carbonic acid, this carbonic acid immediately dissociates into H plus and HCO3 minus. HCO3 minus, that is bicarbonate ions, leave the RBCs, enter into the plasma and combine with the most abundant sodium and potassium present in the plasma to form sodium bicarbonate and potassium bicarbonate. So 70% of carbon dioxide in the deoxygenated blood is now in the form of sodium potassium bicarbonates, that is in the form of bicarbonates. Now 20 to 25% of carbon dioxide combined with hemoglobin. Hemoglobin is free because oxygen has dissociated from it. So the more the oxygen dissociates from hemoglobin, the more it can bind with carbon dioxide. So it binds with hemoglobin to form carb-amino-hemoglobin. Remember, not carboxy. Oxygen binds to form oxyhemoglobin. Carbon dioxide binds to form carb-amino-hemoglobin. So 20 to 25% is carb-amino-hemoglobin, 70% is bicarbonates, and 7% is dissolved form. Now in bicarbonate 70%, 20 to 25% carb-amino-hemoglobin, 7% dissolved form. It enters into the right atrium, from there to the right ventricle, from there deoxygenated blood via pulmonary circulation we know goes to the alveoli. Whatever reaction took place here will all happen in a reverse manner at the alveoli. Binding and dissociation of oxygen with hemoglobin, my dear children, depends upon partial pressure of oxygen mainly. Similarly, binding and dissociation of CO2 with hemoglobin also not just depends upon PCO2, that is partial pressure of carbon dioxide, but please make a note, mainly with respect to partial pressure of oxygen. Yes, partial pressure of oxygen will mainly decide the binding and dissociation of carbon dioxide with hemoglobin. So as oxygen starts to diffuse inside, the HBH plus dissociates, hemoglobin now binds with oxygen and H plus ions are set free. These H plus ions now catalyze NaHCO3 and KHCO3 breakdown to form Na plus K plus, see here, to form, and HCO3 minus. This happened in the reverse direction at the tissues, happening in the reverse direction at the alveoli. H plus and HCO3 minus will now combine together in the presence of carbonic anhydrase, maximally in the RBCs, small amount in the plasma. Also, again, make a note of this, can come as your MCQ, true and false, to form H2CO3, which dissociates into H2O and CO2. And that is the reason when you blow air like this as an expiration process, you also have water vapor. You must have done this in front of the mirror as a child, isn't it? Yes. So that is CO2 and H2O, metabolic water, which is released out. More oxygen entering and diffusing inside from the alveoli into the blood will also dissociate carbaminohemoglobin, releasing carbon dioxide, which is exhaled out and put into the alveolar air, and hemoglobin again free to bind with oxygen. So this is how oxygen is transported. This is how carbon dioxide is transported. I hope this is absolutely clear how the transport takes place. Also, I explained you one very important point which can come as MCQ, that binding of CO2 depends upon partial pressure of carbon dioxide with hemoglobin. However, but main one is partial pressure of oxygen. Similarly, binding of oxygen with hemoglobin is partial pressure of oxygen and also partial pressure of CO2, H plus, and temperature. Now, how do we know that what are the other factors that affect binding of oxygen with hemoglobin? Let us understand it through a graph. This is the NCERT diagram of transport of gases. Why have I given this here when I have explained it beautifully through this diagram? Because this has come as a PYQ. This has come as a PYQ, where they have asked as a labeling of systemic artery, systemic vein, pulmonary artery, pulmonary vein, or they can ask you the partial pressures. Okay? Yeah. So before we move ahead to understanding of what are the other factors that will affect binding of oxygen with hemoglobin, I have just kind of summarized the entire part here. The medium of transport of gases is blood. Via process called, can you quickly tell me, via process called? Via process called diffusion, yes. How is oxygen transported? Oxyhemoglobin 97%, dissolved form 3%. Now again, one more thing that can come in the MCQs is this part. 100 ml of blood delivers 5 ml of oxygen to the tissues under normal physiological activities. Let us understand how. 1 gram of hemoglobin can carry 1.34 ml of oxygen. Remember, 1.34 ml of oxygen. That is, 1 molecule of hemoglobin can combine with or can carry 4 molecules of oxygen. Molecules 4 ml, 1.34 ml. 1 gram of hemoglobin, 1.34 ml. Now in 100 ml of blood, how much is hemoglobin present? Circulation chapter enumerates there are 12 to 16 grams of hemoglobin in 100 ml of blood. Let us take an average 15 grams of hemoglobin in 100 ml. So 15 grams in 100 ml and 1 gram hemoglobin 1.34 ml. So simple mathematics, 100 ml of blood can carry 15 into 1.34 ml. On an average, don't worry, I've done the calculation for you. It is 20.1 ml of oxygen. But ma'am, you just said it delivers 5 ml and here you are saying 20 ml. Now this is a twist that can come wherein they can ask you 100 ml of blood can transport. My dear NEET aspirants, remember 100 ml can transport 20.1 ml but delivers 5 ml under normal physiological conditions. So what if I am doing some kind of an activity, I am running on the treadmill, at that point of time it will deliver more, 15 ml maximum of oxygen it can deliver. But under normal conditions, 100 ml delivers 5 ml. This can again come as our, yes, MCQ. So make a note of it in your NCERT or in the books. Carbon dioxide I have explained, bicarbonate 70%. In the RBCs as carb, aminohemoglobin, make a note of it again, 20 to 25% and dissolved form how much percent? 70%. Again an MCQ. Okay. So, just give me a second, I'll have a sip of water please. Now, I just now told you that other than partial pressure of oxygen, there are other factors that will also decide or also kind of make an impact on how oxygen or how much oxygen binds with hemoglobin. What are the other factors? The other factors are partial pressure of oxygen, partial pressure of CO2, H plus ion concentration, thereby the pH, and also temperature. How do we understand that? Let us first understand the binding of oxygen with hemoglobin. If we draw a graph wherein on one axis we are taking partial pressure of oxygen in mm of Hg and on the other axis the percentage saturation of hemoglobin with oxygen, if we plot a graph, see if I keep on increasing the partial pressure, what do you think will happen? Will oxygen bind with hemoglobin or dissociate with hemoglobin? What will happen? Quickly let me know on the chat. Yes, it will bind. Now as it binds, very good, very good. So as it binds, you see that the hemoglobin keeps on getting saturated with oxygen. So as we keep on increasing the partial pressure, the saturation of hemoglobin with oxygen increases. But it increases up to a particular point, post which we cannot have a graph which keeps on going like this, right? Why not, ma'am? The reason being that after a particular point, all the hemoglobin molecules are saturated, no, with oxygen. Where do you have other free hemoglobin molecules? We don't have. So then, it remains as a plateau, giving us an S-shaped graph, and this graph is called as a sigmoid curve. So if you pay attention to what is given in the theory, that oxygenated blood has 95 mm of Hg of your partial pressure of oxygen, right? We just discussed. So at 95 mm you see almost 100% is saturated. And if you see at approximately 27 mm of Hg, my 50% hemoglobin is saturated, that is P50. What is P50? At 27, at a particular partial pressure, 50% of hemoglobin saturated. Now think and recollect, what did we say? What is the partial pressure of oxygen in deoxygenated blood? 40 mm of Hg. My dear children, see at 40 mm of Hg, almost my 75% hemoglobin is saturated. Is this clear? So as we keep on increasing the partial pressure, the oxygen saturation with hemoglobin keeps on increasing. So what if my partial pressure of oxygen reduces, partial pressure of CO2 increases, thereby H plus ion concentration increase. If H plus increases, pH decreases and temperature increases, my oxygen dissociation curve will shift to the right. And where does this take place? This factor occurs, that is curve shifting to the right, occurs at the level of tissues, again can come as our MCQ. The exact opposite, that if the partial pressure increases, PCO2 of carbon dioxide partial pressure decreases, H plus ion concentration decreases, pH increases and temperature decreases, the curve shifts to the left, and this occurs at the level of alveoli. So with this graph we can easily understand what happens to the diffusion, to the binding of oxygen with hemoglobin if the other factors also vary. Is that clear? Very good. Now, we have discussed in detail about the physiology of transport of gases. Let us quickly go on to understanding of the question. Now, I am bringing questions like assertion, reason and match the column because these are where you tend to make little mistakes. So, let us practice those. Assertion being maximum amount of oxygen is carried bound with hemoglobin. Reason, maximum amount of carbon dioxide is carried as carboxyhemoglobin. Giving you a time of 15 seconds, quickly tell me the answer. Very good, Mr. Gada has given us the answer. Very quick to give the answer, very good. Let me see who else. Okay. Yes, Khushi. Yes, Mrunal. Very good. Yes, very good. All of you all have given the correct answer. Let us quickly have a look at it. Maximum amount of oxygen is carried bound with hemoglobin. Yes, 97% oxyhemoglobin. So this is a true statement. Maximum amount of carbon dioxide is carried as carboxyhemoglobin. No, it is carbaminohemoglobin. So this is a false statement. So assertion is true, reason is false, and which key has our answer? Is the answer key C. Quickly, moving ahead to the next question. Very good, Abhay, very good. Sadhya, very good. Khushi, Mrunal, very good. Chaliye. Again, quickly, in next 15 seconds, be very quick, my excellent, intelligent children. Moving ahead with the next question. Choose the incorrect statement. Every 100 ml of deoxygenated blood carries 4 ml of carbon dioxide. Carbonic anhydrase is present in high concentration in RBCs. High PCO2, low PO2 helps the tissues in binding of carbon dioxide, and oxygen is carried by hemoglobin as aminohemoglobin. Quickly, can I have the answer for this? Mr. Gada again, very good. First one to answer, Mr. Gada, very nice, impressive. Yes, Khushi, Mrunal, very good. Yes, let us see the answer. So here we have to choose an incorrect statement. Every 100 ml of deoxygenated blood carries 4 ml of carbon dioxide. Yes. Carbonic anhydrase present in high concentration in RBCs. Again correct. High PCO2 and low PO2 helps in binding of carbon dioxide. Again yes. Oxygen is carried as carbaminohemoglobin. No, this is the false statement and this is our answer. Very, very good. Very good. Very good. Very good. Chaliye. Moving ahead. So that was about breathing and exchange of gases, conceptual part. Moving ahead to understanding of the heart. Now when we talk about our heart beating, heart is an organ that starts to beat before we were born because it starts beating at 1 month gestational age. We'll study in reproduction chapter, and continues to beat till the person dies. How does this beat and how many times does it beat? It beats on an average 70 to 75 times per minute. That is on an average 72 times per minute. So if it is beating 72 times per minute, means for one heartbeat, it needs 0.8 seconds. Now my dear children, tell me, will every 0.8 second, is it feasible for the brain to send signal again and again and again and again and again to the heart? Ki baba heart beat, 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 beat. Difficult. Our brain will literally get tired. So what has God done is made a heart autorhythmic, auto-excitable, myogenic. Now what do we mean by these terms? Auto-rhythmic, auto-excitable means it has the capacity to generate its own impulse on its own. It's myogenic. Myo means muscle, genic means genesis, production, means it has the capacity to produce its own impulses. How does it do? Now there are certain musculature in the, or certain parts of the musculature of the cardiac muscles itself which get modified to form what is called as the nodal tissue. There is a small patch on the right upper corner of the right atrium. Here, if you see, the right upper corner of the right atrium is a small patch which is called as the SA node. What is it called? The SA node. Now this SA node or the sinoatrial node then is the one which is called as the pacemaker. Why? We'll talk about it in some time. Another patch which is present in the lower left corner circular mass of the right atrium itself called as the AV node. From there arises a small bundle which passes to the atrioventricular septum and towards the upper part. You can see this is the interventricular septum. Towards the upper part of the interventricular septum called as the AV bundle. So we have the AV node, we have the AV bundle. From the AV bundle then arises two branches called right and left bundle branch, and which then continues as the Purkinje fibers present in the musculature of the ventricles. Now this entire tissue is called as the nodal tissue. They are cardiac muscle fibers but which work as nerves. These are the ones which have the capacity to generate its own impulse. Do all of them have the capacity? Yes, all of them have the capacity. Then why is SA node called as the pacemaker? Because SA node, can you think? Can you quickly think and tell me the answer? Anyone? Yes, because what happens is SA node is the fastest to generate its own impulse. So because it is the fastest to generate its own impulse, it will generate the impulse, sending it further ahead, and the impulse is just conducted. Hence, it is the one that sets the pace and hence called as the, or generates the pace or generates the impulse for the heart to beat, hence called as the pacemaker of the heart. Is that clear? So now when it generates the impulse and passes it further ahead, how does the heart actually beat? What happens in the heart during one heartbeat? We know there are 4 chambers. All the 4 chambers need to contract and relax during one heartbeat. So there is a sequential event or series of events that occurs during one heartbeat. Now that series of events that occur during one heartbeat, during once when the all the 4 chambers contract and relax, is called as the cardiac cycle. So here with this diagrammatic representation, I am going to explain you the cardiac cycle. I am going to talk a small glimpse of your ECG also. Before we move ahead to understanding of entire part of this, let me just give you an understanding of when we are talking about ECG, what is it actually? It's an electrocardiogram. It's a graphical recording of the electrical activity of the heart. The heart conducts impulses across its nodal tissue, and these are nothing but minute electric currents, which we will be talking about again when we talk about the neural control chapter. Remember one thing till then is in the muscle, when there is depolarization, when the impulse is being transmitted, there is depolarization, it causes systole, that is contraction of the heart. When there is repolarization, it causes relaxation of the heart. So, what are the 3 main parts which occur during one heartbeat during cardiac cycle is atrial systole, ventricular systole, and joint diastole. What is atrial systole? Atrial systole is the time when the atria contracts, which is approximately 0.1 second. Ventricular systole is 0.3 seconds and joint diastole is 0.4 seconds. Now what happens when all of this occur? Let us see here. We said that SA node generates its impulse which passes through both the atria. When it passes through both the atria, it brings about depolarization of the atria, and I just now told you depolarization means what? Systole or diastole? It is systole. So when it passes through both the atria, it brings about contraction of the atria called atrial systole, which lasts for just 0.1 second. When the atria are contracting, the AV valves are already open. Now you will tell me, how did it open? We'll talk about it when we talk about joint diastole. Be a little patient for some time. We come here and then go back again to atrial systole, okay? So when the atria contracts, AV valves are already open, semilunar valves are closed, and the blood goes from the atria to the ventricles. The amount of blood that flows from the atria to ventricles during atrial systole is increased by 30% during atrial systole. Remember, important MCQ. After 0.1 second, and when that 0.1 second occurs, if you are placing leads on the chest wall and on the wrist and the ankle, we will see that on the ECG we have a positive deflection called as P wave indicating atrial systole. Now by then, the impulse has gone to the AV node, from there to the AV bundle to right and left bundle branches and to Purkinje fibers, carrying the impulse to both the ventricles, bringing about depolarization of the ventricles. Again, depolarization means ventricular systole. Ventricular systole lasting for how much time? 0.3 seconds. When the ventricles begin their contraction with the pressure that it exerts to pump the blood into the major arteries, to prevent the backflow, the AV valves will close. As the AV valves close, it produces the first heart sound called as lub. And majority of the blood passes from the ventricles into the major arteries, that is from the left ventricle into the aorta, from the right ventricle into the pulmonary artery. Now, at the time when the ventricles are contracting, the atria have begun its relaxation. However, the atria have not completed its relaxation. So now 0.4 seconds, what is happening is the ventricles begin their relaxation, the atria complete its relaxation. At this point of time, the atria are already full and are being filled with blood from the major veins that bring the blood, the superior inferior vena cava into the right atrium and the pulmonary veins into the left atrium. Now because the atria are full of blood, ventricles are empty because they are kind of beginning to relax. What happens is, as they begin to relax, there is backflow of blood from the arteries, because of which the semilunar valves close and pressure gradient causes the AV valves to open. As the AV valves open, 70% blood goes from the atria to the ventricles because of difference in the pressure gradient, and semilunar valves closing, producing the second heart sound that is called as dub. The ventricular systole on ECG produces QRS complex and the ventricular relaxation produces T wave. Is that clear? So that is those are the three parts that occur, or the three series of events that occur during one cardiac cycle or during one heartbeat. Is that clear? Now the question comes, can you know the heart rate from the ECG? Yes. How? By counting the QRS complex. The number of times you have QRS complex in 1 minute, that is the time that the heart beats in 1 minute. Also, you have to remember, when does the ventricular systole begin? Immediately after Q. And when does the ventricular systole end? At the end of T wave. Is that clear? So that's an understanding of the electrical activity of the heart depicted on ECG and the series of events occurring in the heart during one heartbeat. Is that clear? Yes. The same thing depicted here, wherein you have atrial systole, atria contracting, ventricular systole. When the ventricles are contracting, atria are being filled by the blood, and joint diastole, where 70% blood goes from the atria to ventricle and all the four chambers are simultaneously relaxing. Let's move ahead to the questioning. Okay, the question says, match the column. On one side we have SA node, ventricular systole, atrial systole, joint diastole. Other side we have the timings and the pacemaker of the heart. Now quickly, let me know the answers. I'm giving you again 15 seconds. Mr. Gada, let us see if you are again first in answering this time. Mrunal is first to answer. Mr. Gada, there he is. Abhay, very good. Okay, let us see. SA node will be, yes, your pacemaker of the heart. So 4. Let us see. Now whenever you have matched the column, be very, very careful. If from A only you get the answer, that's the quickest way to solve it. But always, even if you get the answer from A, as you can get here, go and cross-check your other options in the final NEET exam, so that during stress, if you made any mistake, the others will help you. Okay, that's a small pointer that will help you prevent lose your marks and score marks. Ventricular systole is 0.3 seconds, atrial systole 0.1 second, and joint diastole is 0.4 second, and our answer is D. Very, very, very good. All of you all have given correct answers. Excellent, excellent, excellent. Very good, Anurag. Very good, Abhay, Mr. Gada, and Mrinal. Very, very good. Okay, moving ahead to the next question. Chalo, quickly. Again, you have true and false statement. Giving you a time of 30 seconds quickly. Yeah. Oh, Mr. Gada, very good. Again, first to answer. Is it? Check it again once. Runal, Abhay, Anurag. Yes, Abhay, there you have the answer. Let's see. Okay, let's see. Bicuspid and tricuspid valves are open during joint diastole. Now, what did I say? Joint diastole. Now, I can see answers which are a little incorrect. So, let us go into quick recap of what we've studied. Now, when we spoke about joint diastole, my dear children, I told you atria is relaxing, ventricles are relaxing. At this point of time, the AV valves are open. That is, bicuspid, tricuspid are open, semilunar are closed. So, some of you who have answered have not answered it correctly. So again, during joint diastole, bicuspid, tricuspid, AV valves are open. Yes, only then 70% blood, my dear children, will go from the atria to the ventricles. No, so this is a true statement. And also, when you read your NCRT, this is a direct NCRT statement which I have picked up. During atrial systole, there is increase in the blood flow to the ventricles by 30%. Yes. Ventricular systole increases the ventricular pressure, causing closure of bicuspid, tricuspid valves due to attempted backflow. Yes. As the ventricles relax, that is during ventricular diastole, the ventricular pressure falls, causing closure of semilunar valves, which prevents the backflow of blood. Yes. So the answer will be all of the above. Yes. Very good, Mrunal. So answer is all A, B, C, and D. Answer key D. Okay. So that was about the understanding of how the heart beats. Now in locomotion and movement, we go into understanding. We just now saw how heart beats. So as the heart beats, the heart is contracting, relaxing. Similarly, we have our muscles which are contracting, relaxing. Now how do these muscles contract and relax? Let us see. For that, we first have an understanding of the muscle, and then we go on to the understanding of the contraction and relaxation. Now, when we talk about muscle, if you're talking about skeletal muscle, here we are focusing on skeletal muscle and its understanding. Muscle is a fusiform shaped organ. When we take a cross section, this is how it appears. This is the NCRT diagram. This will make it easier for you to understand, so I've drawn it here. Now, muscle is covered by a connective tissue covering, which is called as epimysium. Yeah, the connective tissue covering called epimysium. Within the epimysium, you see multiple bundles. Now, these are bundles of muscle fibers which are called fasciculi. This is the part where students tend to get confused. Given in the NCRT, I have simplified it. It will become very easy. So, within the epimysium, that is within the muscle itself, you have multiple small bundles of muscle fibers. These bundles are called as fasciculi. Holding the fasciculi, you can see these white part here, this white part is nothing but a connective tissue, a collagenous, common collagenous connective tissue called fascia. Fascia holds all these fasciculi together. These fasciculi are covered again by connective tissue called perimysium, within which you have multiple muscle fibers which are covered by a connective tissue called endomysium. So muscle fibers covered by endomysium, bundles of which come together covered by connective tissue called perimysium, forming a fasciculus. Multiple such fasciculi held together by fascia, and fascia with all the fasciculi covered by a connective tissue called epimysium. Is this clear? Okay. So the same thing we can see here. This is the epimysium with the fasciculi, with the fascia in between having blood vessels, within which is muscle fibers covered by endomysium. Now muscle, if we are talking about, is an organ which is made up of obviously tissues. Tissues are made up of cells, and these muscle cells are called muscle fibers. Muscle fibers which are there are made up of parallelly arranged parts or structures which are called myofilaments or myofibrils. Now these myofibrils basically contain two types of proteins called actin and myosin, which are alternately arranged in such a way that it gives dark light banding pattern, like you have the bands on the zebra. Okay, so dark light, black white, similarly you have dark and light. So let us first understand the actin, the myosin, how it is arranged, what is it exactly, how it is placed into the cell, and then we'll understand what is the functional unit of muscle and how it contracts. So moving ahead to understanding of actin. Now when we are talking about actin, actin is basically a filament. It's a protein filament. You know that muscles are made up of protein, so people keep on drinking protein shakes to build up their body, muscle building, right? So there are two main types, actin filament and myosin filament. Now this actin filament which is there is a polymeric structure. It's a polymeric structure. We studied in the last class polymers are made up of monomers. Now here there are three types of structures or three types of proteins that make up your actin filament. The F-actin, the tropomyosin and the troponin. Now this part, this beaded structure that you can see here, let me use another color for you. This beaded structure that you can see here like a necklace is nothing but your F-actin protein. And this F-actin protein is, as you can see, made up of small beaded structure, and these beads are nothing but G-actin. So F-actin is a polymer of G-actin. It's like a necklace of pearls which is intertwined amongst themselves in the helical format. So there are two F-actins, and along the length of the F-actin we have two filamentous proteins called tropomyosin. And at regular intervals of tropomyosin you can see these triplets. These are three subunits of troponin, which are placed, again MCQ, at regular intervals on what? Actin or tropomyosin? It is placed on tropomyosin. Now beneath the tropomyosin is the binding site for myosin, which we need to mask because they have high affinity for each other. Holding the tropomyosin in its position is troponin, one subunit, and hence we say troponin masks the binding site for myosin on actin, an MCQ important question. So that is about actin. When we talk about myosin, myosin basically is again a polymeric structure which is made up of many monomers called meromyosin. Meromyosin has two parts called heavy meromyosin and light meromyosin. This globular head that we can see here is a protein structure itself made up of an enzyme called ATPase enzyme, and is the globular head, followed by a short arm and a long tail. The short arm and the long tail form the light meromyosin, and the globular head forms the heavy meromyosin. The globular head, as I told you, is an ATPase enzyme itself and has binding site for actin and has binding site for ATP. The head and the short arm project at intervals and at an angle from each other, and we call it as cross arm. Now this is the structure of myosin, meromyosin. This is the NCERT diagram explaining the same thing. Now, how are they arranged? I told you they are arranged alternately, but they're not completely overlapping, they're partially overlapping. So you have, as you can see, this is the actin filament, the myosin filament. So actin, myosin, actin, myosin, actin, myosin alternately placed, and I'm sorry, the actin, just give me a second. Actin is partially overlapping the myosin. So there is a part of myosin which is not overlapped by the actin and is called as the H zone. In the center of the actin filament, there is an elastic band, and this elastic band is called as Z line. It's elastic in nature. And this entire part, so this is one actin, this is another actin, these are two Z lines. Between two subsequent or consequent Z lines, you have what is called as the sarcomere. Sarcomere is the functional unit of the muscle cell. So the entire muscle cell is made up of what? Nothing but sarcomeres. Above, below, in front, behind, on the sides is just sarcomeres. So this part is one sarcomere. From here we have another. From where we have the third sarcomere. So if you see across the Z line, there is this part which has only actin filament, and this is called as I band. So I band gives a lighter appearance and has only actin filament. In the center, this black thing that you can see is the Z line. And this part where you have myosin as well as actin is called as the A band. Okay, and this A band has myosin which is partially overlapped by actin, and this appears darker in color. So you have dark light, dark light, dark light banding pattern. This part is one sarcomere, this is another, this is the third sarcomere. That is how the entire muscle cell is, sarcomere after sarcomere. So how does muscle contraction take place? When you are talking about muscle contraction, it is basically shortening of the muscle, reducing the length of the muscle by reducing the length of the muscle cell. How does that take place? Now muscle is having its stimulus coming from the CNS. We are talking about skeletal muscle. It is voluntary. The impulse comes from the central nervous system to the nerve endings, which releases neurotransmitters at the neuromuscular junction. The neurotransmitters bind with the receptors which are present at the neuromuscular junction. Once this takes place, it causes depolarization of the muscle cell. Once depolarization takes place of the muscle cell, the sarcoplasmic reticulum, which is the storehouse of calcium, releases calcium. Simultaneously, mitochondria releases ATP. Now we've studied that on the meromyosin head is ATP binding site. So ATP comes and binds here. The globular head is ATPase enzyme. It breaks down ATP and releases energy. Simultaneously, calcium comes and binds on troponin, and this tropomyosin slings down, exposing the binding site for myosin on actin. Is that clear? Now once that takes place, using the energy, the cross arm takes its position like this. So the cross arm took the energy from ATP because ATP was broken down into ADP and inorganic phosphate, and the meromyosin head now goes and binds with the actin binding site on the myosin binding site on the actin, forming what is called as the cross bridge. Once the cross bridge is formed, the meromyosin head pivots. This is called pivoting. It rotates. As it rotates, it pulls the actin inwards. Let us see with this diagram. You have actin filament. As the meromyosin head binds and pivots, it pulls the actin towards the center of the A band. This was the A band. It pulls the actin towards the center of the A band. See here and see here. What happens as the actin move towards each other, towards the center of the A band, the H zone keeps on reducing, and when the muscle is maximally contracted, the H zone disappears. You see the I band is reducing, look here, and here the I band is reduced further, so the I band has reduced. So I band reduces, the Z lines are brought closer to each other because the actin filaments are brought towards the center. As the Z lines are closure, bringing or coming close to each other, the length of the sarcomere reduces. As the length of the sarcomeres reduce, all the sarcomeres reduce in length. As all the sarcomeres reduce in length, the muscle cell reduces in length. As the muscle cell shortens in length, the muscle also shortens in length, causing muscle contraction. So your MCQ comes where they will ask you that during muscle contraction, what happens to the A band? See here, the A band is retaining its length. There's nothing happening to the A band. The A band retains its length. The I band reduces. See, everything reduces except A band. Remember it that way. Edge zone disappears, reduces and disappears. Z lines come close to each other. Sarcomere reduces. So again, as I said, everything reduces. Sarcomere reduces. Edge zone disappears. I band reduces. Z lines come close to each other. A band is the only one that retains its length. Again, when it has to relax, it uses ATP. And the meromyosine will use the ATP, it will detach and again come back to its position. Thereby, it detaches, again come back to its position, and the actin filament will slide back. As it slides back, it moves to this direction, and again everything what has happened here reverses. That is how muscle contracts and muscle relaxes, which I have summarized here for you all. Nerve impulse coming at the neuromuscular junction, there is release of calcium ions and ATP from sarcoplasmic reticulum and mitochondria. Binding of calcium to troponin, ATP to meromyosine. Tropomyosine shifts and exposes the binding site for myosin on actin. Cross bridge formation takes place, pivoting of meromyosine head. Cross bridge breaks, reactivation of myosin, and this keeps on repeating till calcium is not pumped back into sarcoplasmic reticulum, and this has come as a PYQ. So be very, very careful. So that is how muscle contraction takes place. I hope the entire procedure of muscle contraction and the structure is absolutely clear. Quickly, let us solve 2 more questions and let's take a test. All of the following lengths are reduced during muscle contraction except I just explained you. Quickly, if you can give me the answer. Taksh, Ubaid, Mr. Gada. Yes, Kushi, very good. Runal, yes, the answer is A band. I just told you everything reduces, A band retains its length. It just does not have any change into it. Okay, quickly. Yes, Abhay, very good. Taksh, very good. Mandar, very good. Kushi, very good, very good. Now, quickly, the second question. Assertion, reason, one of your favorite types of questions. The portion of myofibril between 2 successive Z lines is called sarcomere and is the functional unit of contraction. Reason, during muscle contraction, A band reduces, I band retains its length, thereby reducing the length of sarcomere. Quickly. I don't think should take more than a second also. 10 seconds. Very good, very good. I have answers coming up. Very good, Mr. Gada. Gada is always first one to answer. Quite impressive. Mrunal, very good. Mrunal is also very quick in answering. Abhay, very good. Khushi, Mandar, Taksh, quick, bachas. Yes, Khushi, very good. Yes, let's see. So, the portion of the myofibril between 2 successive Z lines is called sarcomere. Yes, it is called sarcomere and is a functional unit. Absolutely correct, an NCRT statement. So, this is true. During muscle contraction, A band reduces. No. A band, what happens to the A band? A band, yes, retains its length. And I band reduces. I have just flipped the words and this becomes a false statement. So assertion is true and reason is false. Answer key C. Very good, very good, very good. So many C's. Yeah. Very good. Okay, quickly. One more question. Chalo. Match the column H band, sarcomere, A band and I band. Yeah, this sir is back. Very good, Jissa. Now answer. I know you are back, but answer quickly. Mr. Gada, Abhay, very good, very good. Taksh, very good. Khushi. Taksh, Abhay, read, bachas, read carefully. We have H band also and we have A band also. Mrunal, very good, Mrunal. Others, quick recap of what you have answered, quickly. Let us see. H band is the functional unit of skeletal muscle. What is it? It is sarcomere. Where is the sarcomere? Sarcomere is here, B. So that is one. H band having only thick filament. Hold on. Band where myosin is not overlapped by actin. Is this not the most appropriate? So your one should be 3. A band is a band having thick filament. NCRT statement. A band has thick filaments, I band has thin filaments, A band has myosin, I band has actin. Only word is not used. Be very careful. So A band is band with thick filaments and I band is band here. So it will be 3, 1, 2, 4. Which band? 3, 1, 2, 4. Answer key D. Got it? Mr. Gada, yes, very good. Mrunal understood. Abhay, very good. Taksh. Is this clear? Yes. So today we have had a quick recap of understanding and physiology of breathing, of physiology of circulation, how heart beats, how lungs are working, how heart is beating, and how muscles are contracting. So we've covered up the most important concepts of these 3 chapters from where questions are asked repeatedly. After looking at this session and after hearing this session, I hope everything is crystal clear to you all. But read it once again from your NCRT, if possible today itself, and it will become very clear to you. Okay, so bidding bye for today, but see you again with the next session. Till then, take care. Bye. Thank you.